Good afternoon, everybody. I'd like to welcome you to the 306th Brookhaven Lecture. If you could take a minute and read the instructions projected up on the screen so we all know what to do in case a fire or other emergency breaks out in the hall, I would be grateful. Today's lecture is given by Joel Sussman, and its title is Ache in 3D. I say ache because I can't pronounce the chemical or the enzyme that he's going to be talking about, Mysteries Revealed from the Crystal Structure. To introduce Joel Sussman is Bill Studier.
Thank you. It's a pleasure to introduce Joel today. Joel Sussman grew up on Long Island and went off to Cornell to college, where he got his degree in physics, and then on to MIT for a PhD in biophysics with Cyrus Leventhal. For his thesis work, he determined the first three-dimensional structure of an RNA. It turned out to be 2 nucleotides long, but RNA nonetheless. And he's continued his interest in three-dimensional structures of molecules, particularly nucleic acids. He went on to be part of the group who first determined the three-dimensional structure of a transfer RNA, RNA involved in protein synthesis. In 1976, he joined the Weizmann Institute in Israel, where he continued to work on problems in structural biology, particularly three-dimensional structure determination of nucleic acids and also proteins. He's had a long interest in how to visualize three-dimensional structures of macromolecules, of biologically active, important macromolecules. And a year ago, January, he came to Brookhaven to head up the protein data bank here, which is the worldwide repository for three-dimensional structure information for biological macromolecules, and led us in a successful winning of a competition to continue that database for the coming years. The molecule in the title of his lecture is acetylcholinesterase, and I don't want to give you much information about that because obviously that's the topic of Joel's lecture, but when he first came to biology, he gave a beautiful seminar on the structure and function behavior of this molecule with beautiful slides and graphics, and as I said, he's been interested in how to display images of biological macromolecules, and I think we're in for a treat today when he tells us about acetylcholinesterase in 3D, Mysteries Revealed from the Crystal Structure.
Thank you very much, Bill. Can you hear okay? Okay, great. All right, if you read the bulletin, you saw that the talk is a big mishmash, but what I wanted to do was to really give you a feeling in the beginning, what is the protein data bank, very, very briefly, and then to look at one particular protein that Bill described, that I'll talk about, acetylcholinesterase, and relate it to the protein data bank to see how when one examines one structure, in fact, one really has to look at a series of other structures. But then towards the very end of the talk, I want to give you some very recent results of some experiments, some of them performed here at the NSLS on some potential drugs for treating some terrible diseases, as well as some interesting structural studies that we've done on a toxin that can kill you, and you can see how the toxin binds this enzyme. These are just recently have come out. So let's begin with the first slide, please, on both sides. Right. Just to put things in perspective, we're mostly going to be talking about proteins today, but all of the information to code for proteins, of course, is stored in the DNA, which is smart enough to code for its own replication with a number of enzymes that help. Certain regions of the DNA are transcribed into RNA, which are then translated into proteins, and there are enzymes that will convert fragments of RNA back into DNA. In fact, a number of the terrible viruses, such as AIDS and the Ebola virus, have this mechanism to cause some of the trouble. Of course, from the DNA, you get out the products of all of the proteins, and it's still, to me, amazing. This is a picture of my 20-year-old son, but and the DNA was actually larger. This is a real DNA. It's not a computer DNA, and I'll never forget when he came to the lab once. There was an open day at the Weizmann. He came to the lab, and someone showed him a piece of DNA in a test tube. He says, "No, that's not what DNA looks like this." And they showed him real DNA in a test tube. But his younger brother, who's 10 now, looks virtually identical. So physically they're very similar, but the personalities are different. So it's some things come out similar. Right. So could I have the next slide? Next slides, please. The Protein Data Bank was established here at Brookhaven by Walter Hamilton in the early 1970s and then carried on until the end of 1993 by Tom Ketzel, who was head of the PDB. And Walter had a real vision that it would be something very valuable to the whole community if in one site we could collect the structures, the three-dimensional structures of protein molecules, store them, and then disseminate them freely to anyone who wanted them. And this was not available until he did this, and he had great recognition throughout the whole international community and was able to establish it here. In the beginning, there weren't many structures, and a lot of effort was spent in trying to get people to deposit them. And you can see for some period the number of structures was increasing linearly. And then towards the middle of 1980s, there was an enormous increase in the number of structures, almost an explosion. And part of the reasons are listed on this slide over here. Probably the most important reason are the real advances in molecular biology that allow scientists to decide which proteins they'd like to study by cloning the DNA and then expressing that DNA, usually in prokaryotic systems, and getting out basically the proteins they would like. One of the problems is some of these proteins don't crystallize. So a routine way of crystallizing them now is to make a tiny mutant that doesn't affect the function, but somehow helps the protein crystallize. So this is probably the most important event in terms of crystallography, the connection with molecular biology. There are much faster and better X-ray detectors, better X-ray sources, especially synchrotrons, which allow scientists to work with extremely tiny crystals that are down to 10 microns or even smaller, computer graphics and better computers. And finally, the advent of structure determination, not just by X-ray crystallography, but by NMR. So this part which comes out in red over here, these are the number of NMR structures that are available. And as of middle of May, there are 3,531 different sets of coordinates in the protein data bank. Next pair of slides, please. One of the other things which has happened in the last year or so is that it's become much, much easier to access the protein data bank. We've set up a home web page, www.pdb.bnl.gov, where one can very easily access the information, especially going to a browser that was developed by Enrique Abola and Dave Stampf, and it's shown over here. When one clicks on this, a page opens up on your PC, Macintosh, or Unix workstation, which allows you to ask questions if you wanted to find all structures by a particular author. For example, the author Harel, you would type Harel, send this request, it would go over the internet and come back with a list of hits. You would click on here and suddenly get a three-dimensional image of the structure. It's not just a static image. These little bars here allow you to rotate the structure. You just put your mouse in the middle and drag the mouse around. And this same picture of my son, who really doesn't like computers, came to the lab one time and noticed what I was doing and just couldn't believe this. And so he sat down and began looking at protein structures and said, could he print a picture? He printed a picture. And he had never really played with these computers. The next day he called, which was unusual, he called the lab and said, could he bring a friend to show the friend what was happening? So this just came out in a recent article we published in Nature describing this browser. But it's possible to ask a lot of other questions, to get the newsletters and frequently asked questions, as well as pointers to a lot of other databases, including magnetic NMR databases and so on, from this web. In fact, over here, there's actually an entire course on protein structures being given at Birkbeck College, and they've allowed us to make a mirror copy of this course. So if anyone wants to learn principles of protein structure, one clicks on that and can study it and actually get an exam at the end, if you like. Next slide. And just I want to mention the people who have done all this work. This is the really incredible staff at the PDB who are listed here, and I want to give special credit to Dave Stumpf and Enrique Abola, who have really done an enormously good job. And recently, Chaim Preluski, who has joined our group as the interim head of database, is helping to convert these flat files into a full relational database to make it easier to search. And this is the current staff of the PDB, many of the people who are here. All right, next. And this is just over here. I'll just dim it so you can see it better, is one of the images, in addition to these three-dimensional images that can rotate, in collaboration with a colleague in Geneva, Manuel Pech. He's made a collection of most of the protein structures where he's tried to give some glimpse of the important part of the experiment. And this is actually a close-up of the molecule I'll be describing, acetylcholinesterase, showing the active site of the enzyme and where the substrate binds, surrounded by all of these pinkish-colored residues, which are aromatic residues, which I'll talk more about. But for most of the structures in the data bank, he has made really very beautiful pictures, which are also available through the World Wide Web. Next slides. So this gives you some background about what is in the PDB, but I thought it would be worthwhile now to go into some detail about this one structure and to see how it relates to the other structures in the data bank. I'm going to talk about AIC, acetylcholinesterase, which is an enzyme involved in breaking down a very small molecule, which is the neurotransmitter acetylcholine. When a nerve signal comes shooting down a nerve, it causes a neurotransmitter to be released and sent across the synapse. In fact, two nerve cells don't communicate by electrical impulses, they actually communicate by a chemical impulse. So the chemical impulse goes across this synapse, lodges in a receptor, transmits a signal to the surface of the membrane on the second side, which then results in a depolarization of the membrane and then causes a nerve signal to be propagated. And this is the same for the nerve-muscle junction. It works with these neurotransmitters so that when a nerve impulse comes down, goes across this distance, which is about 400 or 500 angstroms. When it gets here, it causes the muscle to twitch. In order for it to stop twitching, this neurotransmitter has to be broken down sort of to set the whole thing back to zero to start again. And that's the function of this enzyme. It hydrolyzes the neurotransmitter to allow the whole process to begin again. We were interested in studying this just to basically understand the whole process of how this enzyme works. It works very rapidly. It can hydrolyze 25,000 neurotransmitters per second, which allows up to about 1,000 nerve impulses to be transmitted per second. It's extremely specific for acetylcholine. It won't hydrolyze, for example, butyrylcholine. And third reason we were interested is for a number of disorders. People have been interested in seeing if it's possible to mediate the action of this enzyme, in particular for one terrible disease, Alzheimer's disease. It's known that there's a deficiency of acetylcholine in the brain, and one of the approaches to just maintenance of the disease, to try to improve some cognitive functions, not really to cure the disease, but just to slow down the effect, would be to moderate the enzyme by making some moderate inhibitor. Of course, if you use a terrible inhibitor that binds covalently to this enzyme, you will be killed. And in fact, I guess everyone saw this horrible thing that happened in Tokyo, where sarin was used in the subway system. Sarin binds covalently to this enzyme, and when you really get a strong dose of it, it affects your respiratory system because the neurosignals don't go to your respiratory system, you die very rapidly. The source of the enzyme we chose wasn't cloned, but it was from a fish, a torpedo, which really grows up to a meter. This is a baby torpedo. In this region of the torpedo, the electric organ is very rich in both the enzyme as well as this receptor. So we can isolate from one torpedo 10 to 15 milligrams of very pure material. Next slide. Next slide's good. And the enzyme is a homopolymer consisting of 537 residues represented by this circle, connected to another molecule identical to that with a covalent linkage in its last residue. And these pairs of molecules are attached to a membrane by a sugar phosphate linkage, a phosphatidylinositol linkage. And the enzyme was purified by cleaving this portion with a particular lipase, a bacterial lipase, that freed this homodimer into solution, and it was crystallized. When one looks at a close-up of what was known about the enzyme before the X-ray structure was determined, it was seen that the binding of the substrate, the acetylcholine, which is shown here, appeared to bind in the active site in two regions. One, the anionic site, the AS, where the choline moiety bound, which is a positively charged group, N plus with 3 methyl groups surrounding it. And this region here, the asteric site where the actual catalysis occurred, this bond is being broken. And that appeared to be rather normal, consisting of a mechanism similar to other hydrolases of a histidine, a serine, and some acidic group, which was not known at the time. This serine is the serine where all these terrible, terrible agents bind, and once it binds here, it stops the enzyme. There was a second site of binding, which from kinetic studies appears to be non-competitive, called the peripheral anionic site, and work of Palmer Taylor by means of fluorescent measurements indicated that the main site and this peripheral site was about 20 angstroms apart, but no one knew much about that. And towards the very end of the talk, when we get to this snake venom toxin, we'll see it in fact binds up here, and you'll see how binding 18 angstroms away from the active site can still easily inhibit this enzyme and kill you. Right. Next slides. The way in which we did this work is by growing crystals, and growing crystals is still a difficult task. One has to set up a number of different conditions and look at them very carefully until you find some that crystallize. And one usually does that by putting a drop of a protein solution on a cover slip of a microscope, usually about 5 microliters. And one puts it down like this and then turns it upside down. It usually doesn't drop out. And one puts it on top of a well where the well contains some solution of a precipitating agent. And then one puts a bit of vacuum grease here. When one covers up the slide on the vacuum grease, it all seals and it's a closed environment. And slowly, some of this precipitating agent, for example, ethanol, would evaporate into the drop, and over the course of a day or two, you can get crystals. And this is a typical crystal which we obtained from acetylcholinesterase. It grows up to almost 1 millimeter in length. You can see it with your naked eye, and it withstands X-rays very, very well. Next slides. X-ray crystallography is exactly the same physics precisely as light microscopy, where you just have light coming in to your specimen. It scatters, and you have a lens that focuses it, and you can see it. And so X-ray crystallography is the same, just the wavelength is somewhat shorter. This is 5,000 angstroms, and this is about 1 angstrom wavelength. Hits your specimen, scatters in all directions. The only problem is that there's no X-ray lens yet. And so in order to focus the scattered rays, you still need a crystallographer. and the computer helps. And with the computer and crystallographer, in some ways to get out information that is inherently lost in the experiment. And the information that's lost is the phase information. When you record the images of the X-ray diffraction, you just record the amplitudes of the scattered rays in the actual phase of the relative phase of when the different rays hit the film is lost. This can be obtained back by a number of ways, which I won't go into today, but using a synchrotron can greatly facilitate getting that information back. But if one can get the phases, which is done by a number of tricks, together with the crystallographer, one can then reconstruct, in essence, make a lens and see an image of what's scattering the X-rays. And what's scattering the X-rays are the electrons that surround the nucleus. So you get out an image of an electron density map of the molecule. And from that you can then build what the structure looks like into that map. And it's very important to realize that the image you're seeing is not a model, but it's actually an ab initio determination of the structure from the experiment. And this is distinct from just trying to predict the structure from let's say, the sequence. Next slides. This is just a picture of setup with some of the work we've been doing here. This is Bob Sweet down here, who has been a very good collaborator. And this is one of the setups where the crystal would sit at the tip of this goniometer head. It's bathed in boiled liquid nitrogen, which can freeze the crystal and allow it to diffract more or less indefinitely in the X-ray beam. And if you don't do this for many protein crystals in the synchrotron, it just evaporates. And this is a typical image. This image was actually taken on a home rotating anode source in about 180 seconds. On a synchrotron, it could be taken in a few seconds. And from this image, one takes a series of images, rotating the crystal until you get a full diffraction pattern. Next slide. And then from that, and with the number of tricks of the crystallographers, and these are two of my colleagues, Michal Arel and Felix Frollo, who were originators in this project with me, together with Israel Sussman, we got out our first maps of this while I was still back in Israel, showing the map of a 60 angstrom projection, sort of like in a butcher shop where you project a series of slices of salami onto one sheet of paper, 60 angstroms thick. And this distance from here to here is 60 angstroms, and this is 60 angstroms. And what you can see is this is the electron density corresponding to one monomer of acetylcholinesterase, and this is the electron density for the second, and this is where they're held together at the C terminal. And most of this other less electron dense region corresponds to the solvent. In fact, there's almost 60% solvent in this unit cell. So sometimes people wonder, is there any relationship at all between the structure you see in a solid state and in solution? In fact, there's a lot of relationship because, in fact, there's very, very few points where one protein molecule actually interacts with the second one. In fact, it's still amazing in some sense that they crystallize this way. It's in some sense so fragile. So it's really mostly interacting with solvent. Next slides. This is a picture of the homodimer over here in a close-up of the region of the active site. The active sites are somewhere buried deep in the enzyme here, and the arrows are just pointing sort of how we think the substrate must go in and here and here. This is the active site serine, which actually forms a temporary covalent linkage to the acetylcholine during catalysis. And what was surprising is that some of the studies before we did this crystal structure indicated that the region surrounding the active site should be filled with a number of negatively charged groups to attract the positively charged choline moiety. And in fact, what we saw are a series of aromatic residues, not negatively charged groups. And this is actually looking into the active site, sort of looking, if your eye were here, looking down this way, looking into it. And if one goes towards you, going out, the whole entrance to the active site is just coated with aromatic residues. That was shown in that color picture that I showed earlier on by Manuel Petch. This was a real surprise to us, and a surprise to a lot of the people in the field. It wasn't a surprise to small molecule crystallographers, and a man named Marcel Verdonk from Utrecht did a study of a collection of small crystal structures, small organic structures, and it turns out it's very common for positively charged groups, especially choline moieties, to be nestled up next to aromatic residues. So this turned out to be well known, but not to protein crystallographers. Now it turns out to appear in lots of protein structures as well, that you see this kind of interaction of a choline moiety near the an aromatic residue. Next slides. But what was really even more surprising for us was the overall structure. And this is shown in a schematic image here where the green arrows correspond to portions of the protein structure which are most extended, where if you just stretch out the structure, it's in almost a straight chain, and the red portions are small alpha helical regions. This is a trace of the backbone of this long linear chain of acetylcholinesterase, the 537 amino acids, as it's folded up in what we saw in the crystal structure. About the same time, a structure of a totally different enzyme, an enzyme from a fungus, Geotrichum candidum fungus, which is and this was a lipase enzyme, was determined in Montreal in the laboratory of Merrick Sigler. And if one compares these two, there are 14 beta strands across this region and across this region. In cholinesterase, there are there's 11 beta strands here. There's 14 alpha helices up here, 15 alpha helices, but most of the alpha helices line up the same way. And it turns out these two structures, when you compare the positions of the central atom of each amino acid, the carbon alpha, they line up to be within about 1.4 angstroms of each other, the RMS deviation of these two when you superimpose them. They are more similar, these two proteins that have virtually no sequence similarity, than the same protein determined in two different laboratories. And this was a real surprise to us, that an enzyme which cleaves acetylcholine very rapidly, the other one which cleaves a triglyceride, would have a similar fold. Next slides. But it turned out that wasn't the end. About the same time that this lipase was determined by Merrick, there was a carboxypeptidase from wheat, a dialactone hydrolase from Pseudomonas, and a haloalkane dehalogenase from another Xenotrippus atropicus, determined in 3 different laboratories in Oregon, in I don't know where Dave was at the time, now he's in Australia, and in Holland. These 5 proteins that really share virtually no sequence relationship, except a tiny little bit between these two, but the rest of them, if you were to compare the sequences of the amino acids, there's virtually nothing in common, all share this very similar three-dimensional fold, where there's a basic core of 8 strands of extended structure lined this way, with the first strand here, the second going anti-parallel, going across to the third, back to the fourth, fifth, sixth, seventh, and eighth. The alpha helices lie in the same position for these five structures, and the active site residues, the residues that are involved in catalysis, these little green balls, lie at the same location in the topology, at the end of the 5th, end of the 7th, and between the 8th strand and the 8th helix. So it appears that this is an example of a series of protein structures that probably evolved from some common primordial ancestor many, many years ago. This ancestor developed as a very nice fold with the catalytic triad positioned at the ends of these beta strands, lying in a convenient way. Once this was established, the amino acid sequences diverged, but the structures remained the same. So in some sense, these 5 proteins are almost imitation for people who are interested to try to predict structure. We have cases of 5 proteins with virtually no relationship in the sequence folding up into the same way. And this problem of trying to predict the structure of proteins is really very important, because nowadays, as we heard from the talk of Bill Studio, I guess it was 2 months ago, new methods are being developed to very rapidly sequence DNA. From that, it's easy on the computer to translate the sequences into what proteins would be produced. It would be wonderful if one could say what those proteins look like, and even more important, how they function. But right now, there are no real good tools given a sequence to predict the structure. So these examples of where one has a number of different protein structures with different sequences begin to help help give information to try to answer that question. Next slides. So this was one real surprise that came out. This is another picture of the structure of acetylcholinesterase shown as a space-filling model with all the atoms shown. This green patches, these green patches, correspond to aromatic patches throughout the structure. These are benzene-like rings. And this entrance to the active site is just covered with aromatic residues. This red dot in the middle corresponds to the serine, this serine residue over there, serine 200. And one can see, even though it's a mono picture, that this is probably the way the substrate would go into this active site. And it was not at all obvious. And this is one of the mysteries that I try to put in the title. What would attract this substrate to go down this long route? It's 20 angstroms almost in the middle of the protein. Why would it go down there to be hydrolyzed? And how in the world would it come out to be taking place close to diffusion limited rate? So what we did is, when we determined the structure, we took the liberty and built a model of how we thought the transition state of the acetylcholine bound in the active site. This was based on some previous studies we had done in chymotrypsin, where we saw bound substrates or bound inhibitors forming a temporary covalent bond to serine 200. This carbon and oxygen interacts with 3 nitrogen groups here to hold it in place, and the full chain of the acetylcholine is extended facing up to tryptophan 84 over here. And there was a great deal of chemical and mutagenic studies indicating that this particular tryptophan was involved in binding to the choline. But this was just a prediction. And the way we thought to study this would be to try to bind inhibitors that look like acetylcholine and see if it bound in the active site anything like this. Next slides. And these are some of the inhibitors we looked at. Edrophonium, which is used for diagnostic purposes in myasthenia gravis, it has a quaternary group. This decanothonium, which was used in purification of the enzyme. Tacrine, which has only a tertiary group here, is the first drug available in the United States for the treatment of the early onset of Alzheimer's disease. And this, we were curious how this bound. It's known that the target is acetylcholinesterase, and we wanted to see how it was bound. And this last molecule, which really looks completely different, it's hard to imagine this thing would bind, considering all the others, is called huperzine. It's an herb. It's a herb from a leaf of a moss found in China, which has been used for over 1,000 years as kind of a folk medicine that was given to elderly people who had lost their memory. And if they drank a little bit of this tea, their memory returned. And this turns out to have 2 orders of magnitude a higher affinity to acetylcholinesterase than tacrine. And it's thought now, since it's a natural product and has been used for so long, a number of people are interested in trying to test this as a possible way of treating the early onset of Alzheimer's. So I'll show you the structures of these 3 in stereo. This is the first one. For people who can see stereo, you can just sort of let your eyes relax and see it in 3-dimension. Edrophonium with this quaternary group nestled up right next to tryptophan 84. We see 2 water molecules in the crystal structure, and it's clear how this would interfere with the active site because acetylcholine couldn't get into the active site. Next slides. This is showing tacrine. It binds in a very similar place, a little bit to your left, closer to tryptophan 84, and a second aromatic group comes down and covers this tacrine. And this is just, it's not an ad, just showing that it really is being advertised for this. On the back side, you can read all the warnings about it and so on, but I only show you the front of this advertisement right now. So it binds in a similar way. This phenylalanine 330 turns out to be rather flexible, and it moved quite a bit between the edrophonium and tacrine. And the next slides show the huperzine structure. And this is work that we just completed. Huperzine also binds about in the same place in the active site. You can see how the electron density map for the difference Fourier in this case really fits very nicely to the huperzine. It sits again near this the tryptophan, the phenylalanine, and right near the active site. But what was interesting for me, when I looked at this structure, we went back and placed on the electron density that we see for the huperzine where waters are found in the active site in the native enzyme. And we see 2, 4, 6 water molecules. So the water molecules themselves almost form kind of a ghost image of huperzine. And we think this may be part of the reason why huperzine binds so tightly, because in fact it releases these waters which have sort of set up a pocket for the huperzine to go in. Once these waters are released into the solution, there's obviously entropic advantage for that, so that is gained. So the binding is not just because of nice interaction with the residues here, but in some sense because of the release of the water. Because when we look at this huperzine, to our amazement, although the binding constant is close to 10 to the minus 9, there's only one direct interaction with the protein between the end of this carbon oxygen and a tyrosine residue. All of the rest of the interactions are mediated through waters or hydrophobic interactions with some of these aromatic residues. And this was a real surprise to us until we saw that maybe it's due to the release of the water. Next slides. So these are just some of the references to articles that we published on this. This article here by Harel et al. Describes the binding of these three structures. And this article by Olis et al. And Adrian Goldman compares the different protein structures. This is a picture of the active site gorge leading into the enzyme's bottom. Most of the residues of the protein hardly change at all on binding the different ligands. This serine stays in place, the tryptophan stays in place, another tryptophan up near the top hardly moves. Only this one residue, phenylalanine, moves from here, binding to the tacrin, here, binding to edrophonium, and I didn't show this decamethonium, but it binds this way to decamethonium. In some sense, it's kind of a swinging gate that helps lock in the different substrates. And we found once we did these structures, it's now possible to think of redesigning a molecule like a modified version of this tacrin that might bind as well here, but would have some other group that would eliminate some of the potential side effects of this drug. So anyway, this is the work showing the various drugs binding to the active site. We were still very curious what attracted these down to the bottom of this gorge. Next slides. And there was some work done by two young theoretical chemists, Daniel Ripoll and Carlos Fairmont, who are at Cornell University. And I gave a lecture about 2 years ago, and they were in the audience, and they asked for the coordinates, and I gave them the coordinates, and within a week they sent me back this picture. And this picture shows the active site lined up over here. All of the red mesh that's on top, this red net, corresponds to negative electrostatic potential. The blue is positive electrostatic potential. The whole molecule turns out to be an enormous electric dipole. This dipole, which is shown by this arrow, is close to 500 dBi, and we've subsequently, in collaboration with Dietmar Porsche in Göttingen, measured this in solution. In fact, the measured values are about twice as large as the calculated one. It's one of the largest electric dipoles of any protein that we know of. If one looks at it in terms of not just the potential, but in terms of electric field, vectors, these little green vectors correspond to vectors that would all be pointing into this cutaway picture of the active site gorge, all leading down to the bottom of the gorge where the serine and tryptophan are down here to attract the substrate. So what we think now is that the very fast on rate for the substrates is in part due to the attraction because of the unusual distribution of charges. The charges aren't lined up in the gorge, but in fact they're distributed all across the northern hemisphere up here. They're mostly acidic groups, and the larger percent of arginines and lysine, the basic groups, are in the southern hemisphere. So this sort of gestalt distribution of charges makes this polarization, which would then attract the substrate. This next slides was studied by another colleague, Michael Gilson, who's now at the Center for Advanced Research in Biotechnology in Maryland, where he approached this by doing a series of computer studies of simulating the movement of the protein as a function of time by solving Newton's laws of mechanics in very, very short intervals of time, letting the protein basically relax and seeing the movement of the structure. And when he looked at the structure, initially showing electric field vectors in the static crystal structure, most of the electric field vectors pointed into the active site with these blue residues indicating the aromatic residues surrounding the active site. There was a bit of electric field vectors going in somewhere over here near this tryptophan 4. He let the protein, which was surrounded by a large bath of water in the computer, he let this go for about 125 picoseconds. And after 23 picoseconds, he saw a 1.5 picosecond period of time where there was a second opening right over here corresponding to this region near tryptophan 84. And Michael dubbed this back door. And what we would like to speculate, and this is real speculation now, is that the substrate would go in, be hydrolyzed, and then another substrate would come in and somehow kick out the product through the back door. And so the whole enzyme would work as a production line. And this is not a very usual way that most enzymes have the reaction first near the surface, things come in, go out, come in, go out. So this would be a different kind of mechanism, and we try to think of some ways to test this. Next slide. Just to show you what it would look like, if this is the top of the gorge again, where T is the top, substrate would come in. It's actually a very arduous route. If one tries to build a model of acetylcholine going in, right over here, it really can't squeeze in. It has to breathe. But down here in the simulation, 3 residues went in different directions, and this would be the opening for sort of this back door. And this was written up in an article last year in Science. We tried some site-directed mutagenesis studies, and they're still questionable, the results. So far, those have not yet confirmed our theory. But what we thought would be nice is if we could try to actually see this happen. It would be nice if you could think of doing an experiment, sort of like going into a discotheque and turning on a series of flashes while people are dancing, and you could see them move. And one way to do that experiment is with a synchrotron, whereby if one could put a molecule which could be bound in the active site, get it in there, make sure it's in there, you could study that leisurely by normal crystallography that some inhibitor is bound, and then if it would be possible in the crystal to release that inhibitor in all of the different copies of the molecules in the crystal lattice simultaneously, for example, by flashing it with a laser. And simultaneously, when you flash it with a laser, if it would be possible to collect the entire set of X-ray data in a fraction, in a millisecond, let's say, one could think of then doing the experiment several times where one would flash with the laser and then open the shutter of the where the X-rays are coming out several milliseconds after stimulating with a laser, so that one could follow the off reaction of some of these products. People have tried this here at the lab. Bob Sweet and Paul Singer have done a series of experiments where they've been able to follow water molecules escaping from chymotrypsin. Louise Johnson and Bob Frederick and others have looked at phosphorylase using several techniques. This is somewhat more difficult because of the speed of the reaction. The size is about the size of the diameter is about the size of the phosphorylase, so it's doable. So last summer we began to do preliminary experiments to see first if we could even collect the data in a fraction of a time. To do that, one has to use a technique known as Laue crystallography, where one doesn't just work with monochromatic radiation, but radiation with a wide series of wavelengths. Next slides. And this is shown just some of the summary. If you didn't listen, you can see the summary over here. But this is showing the lab, and just something over here is the synchrotron, out of focus. You can focus a little bit. And we did some experiments on a beamline X26 last summer, together with Paul Singer, and Bob Sweet, and Vivian Stojanov. Next slide. Next slides. And these are just showing the first successful experiments we did where we took a crystal of acetylcholinesterase, and this is now before doing any of the laser work, just to see if we could collect data in a short time. We collected this frame in 4 milliseconds of time. This shows the actual experiment of the spots, and over here are some computer simulations indexing these spots, because of course it's very difficult to index it once you have spots coming from different radiation, different wavelengths. And these fall very, very close, so it was possible to index these. And this was work done by a young graduate student from Utrecht named Raymond Ravelli, and a student, Mia Ravis, also from Utrecht, and the Weitzman, who have now been able to take this data and actually calculate electron density maps that are roughly of the quality of the ones I showed you previously. In parallel, together with the group in Strasbourg, led by Maurice Goldner, he's designed a series of compounds, cage compounds, that will bind in the active site and will be photoreleased. This summer, we're going to be trying the first experiments here to put these cage compounds in to see if we can see them, and hopefully by the end of the summer, begin to do some experiments of trying to release these compounds together with the laser at different steps. Okay, the very, very end, I just want to just tell you of some really brand new results. As you remember, I mentioned that there were 2 main binding sites. This one site at the bottom of the enzyme called the anionic site, and a second site about 18 angstroms away. Next 2 slides. And this picture of the green mamba snake, in this, you don't want to go near that snake if you can avoid it. This snake has a molecule in its venom known as fasciculin, and it's typical of a number of toxins which have 3 fingers sticking out. These are toxins, most of them for acetylcholinesterase. There are other toxins that look very similar to this for the receptor, acetylcholine receptor, as well as ones that affect muscle, cardiotoxins, but they all have very similar kind of structure. No one has been able to, there are about, I would say, 6 or 7 of these now deposited in the protein data bank, and this one was taken from the data bank. It's 1-FAS structure. This particular toxin binds very tightly to acetylcholinesterase, and this is presumably how the snake venom kills you. Once it binds, it kills you very much like these other kinds of toxins, except it's not binding to the active site based on a number of cross-linking studies that were done, mostly in the laboratory of Palmer Taylor, as well as site-directed mutagenic studies. We were able to grow crystals of this toxin together with acetylcholinesterase, and last summer we collected data on beamline X12C. These crystals didn't diffract well on a home rotating anode. We had to work on the beamline X12C, Bob Sweet's beamline, and we recently were able to determine the structure, and these are just really new results, so the slides are unfortunately not quite as good. Next 2 pictures. Right, this is a picture of the surface of acetylcholinesterase, just the accessible surface, showing also this gorge deep into the enzyme. And over here is a picture of what the complex looks like when fasciculin binds, which is shown in green. It binds over here, and once it binds, all of the entrance to the active site is lost. This region up here, in fact, has been identified from a number of other studies to be near this peripheral site, and it's clear now that this fasciculum is doing its dirty work not by forming a covalent bond with the active site residue at the bottom of the gorge, but blocking the entrance into the active site. And so it binds very tightly, and this is I presume the way it does. What's interesting also is it's not having its fingers delve deep into the hole, but in fact it covers the surface very much like the way large antibodies interact with proteins, such as the complex of FAB fragments with lysozyme. So these are the first pictures we've seen, and the structure has now been refined very well, and we are examining in detail the interactions of the various residues. What's interesting is the fasciculum itself has a dipole, and the dipoles line up very well. There's about a 40-degree angle between them, but they're pointing in the same direction, which is in some sense probably what attracts the fasciculum to this enzyme. Next slides. All right, in closing, I don't want to go over all the collaborators. The project really began with the 3 people I showed you in the beginning, together with my colleague Israel Silman, who spent last summer here. But a number of other people have joined working with us in various areas of the project. And I just want to mention Raymond Ravelli again, who did this wonderful work on the Laue studies. Here at Brookhaven, I'm collaborating with Helen Casilla, Vivian Stojanoff, and Bob Sweet, Paul Singer, and Malcolm Keppel. Helen has grown some very, very nice crystals now of various new forms of acetylcholinesterase, including extremely tiny ones still, of crystals of the human enzyme itself. Turns out the human enzyme and the torpedo are closely related in sequence, but the differences are still intriguing, especially, for example, the binding of huperzine seems to bind much more tightly to the human than to the torpedo. And so we're terribly interested in studying this. I actually learned about the huperzine from colleagues at the Mayo Clinic, a young scientist named Y.P. Pang, who built some models of how huperzine fitted into the active site. And it turns out his models are incredibly accurate. We are also collaborating, I mentioned, with the group in Strasbourg who have designed these new compounds for doing the Laue study. The last thing I just want to mention, and then maybe if we have time, I'll just show a very, very short video to help see this. What we've been studying is this enzyme, which is a bit shaped like a Pac-Man, and one can look at the substrate coming in and interacting with the receptor and causing you to twitch like crazy. And what the enzyme does is it obviously hydrolyzes the substrate and puts you back into a relaxed state for the drinks and stuff. We're now beginning also to work on the receptor, and a young postdoc from Oxford is going to be coming here this summer, and he has been working on purified receptor, and he's looking initially now at one fragment of this receptor which actually binds the substrate, and we hope to see that in the near future. Great. Maybe just, if it's okay, I just will show a 3 or 4 minute video which will, I think, help you see the structure. If I can figure out how to work it. Good. This movie was made by Daniel Ripoll. I don't know what that ox means, but don't worry about it. At Cornell University, at the Cornell Theory Center, and it was made with software that's used for studying the surface of Venus. It's not regular software for looking at molecules. This is opening up the structure and showing the active site surrounded by all of these aromatic residues. And in a little bit, you're going to see how the structure really is divided up into a positive and negative region. So this is the acetylcholine to give you some idea of the scale of the substrate. Now what Daniel has done is colored all of the surface regions that are negatively charged red, electrostatic negative potential, and the positive region, greenish, turquoise, or almost white, the way it came out. This is contoured at a very high contour level, and then he'll rotate it around. As he rotates it, he's going to lower the contour level to 1 kilocalorie per mole. As it comes around, you'll see in a second, right about here was the entrance to the active site. This is the negative region, the positive region. It's really quite dramatic, I think, in this video that he made, showing this dichotomy of charge. There are other enzymes like this. Superoxide dismutase also has a large electric dipole, but it's only 100 debis. Well, this is about 5 times larger. So you'll rotate it around, and then in a little bit, he's going to give you a feeling of what it would be like if you were a positive charged substrate going near this structure. But as it rotates around, you'll see in a second, this is where the active site is. And it's really quite clear. And this is showing both this structure of the topology of the protein with the surface displayed as kind of a cloud surrounding it. What's nice is this whole video is also available on the internet. and you can just download it and see it on a PC or at home if you like. So now at the very end, 1 second more, what he's showing is a calculation of what would happen to positively charged ions if they came near this protein. They just be sucked in. There's a real question once they get sucked in where they go out. And this is what made him and us think that maybe something is special happening in this case. So first, this gives you a picture of what would happen if you were not charged. You'd be floating around looking all over for some entrance to get into the enzyme. And then you pan back, and then you'll get the experience of what it would be like if you were acetylcholine just before you were hydrolyzed. So you come on in, and as you go down, you'll feel the surface of all these aromatic groups, which we think are used in part for selecting acetylcholine opposed to other positively charged groups. Then you get down to the bottom of this deep gorge, that's the positive group, and you look up, and that's what you see coming at you. And it would be difficult to think about going back up that way. We're hoping to have a high-speed link to Cornell soon, where we'll be able to actually see these movies being made in real time, back and forth with some of the techniques they've developed. Just 1 second, I'll see who else is acknowledged. The film was made in large part by Richard Gilliland, who works with Daniel Ripoll, and he's really a very talented chemist and a visualizer of molecules. Okay, thank you very much.
I believe we have time for a few questions. Joel, why don't you select the questioners?
No questions. Nick.
In you work at the light source, how long does it take you to take data to get a structure?
The question is, at the light source, how long does it take how many hours does it take? Right. For this fasciculum work, which we did at X12C, it took us about 12 to 15 hours to collect the data, because these crystals just didn't diffract very well. For this other one, the Laue work, it took 4 milliseconds, but that's because it's white radiation and these are well-diffracting crystals. Just parenthetically, I had a chance to look at these crystals on a third-generation synchrotron in Grenoble. We were able to collect an entire data set in 1 microsecond, which is really kind of mind-boggling. Yes?
On the homology of the fold that you've got, the common fold, how does that compare on the homology with the myoglobin fold?
The question is the comparison of the homology of the folds that I described to that of to the myoglobin. You mean, does this look like?
Is this more or less homologous?
Right. This protein doesn't really have much similarity to myoglobin at all. All right. All right.
Between all the myoglobins, there's a lot of homology. Is the homology a lot different?
Okay, no, in that instance, John, the homology that I showed for those 5 proteins is really at about the kind of level that you see in myoglobins, where many of the myoglobins and hemoglobin folds have a functional relationship. Those are very similar. These apparently, the only similarity they have is that they're all, in some sense, hydrolyzed something. But the basic core of those 8 strands of beta structure have a similar kind of similarity to each other as the myoglobin folds. I mean, you would immediately spot that those are similar structures, okay? In fact, the fact is, John, when I looked in the literature, we saw this carboxypeptidase published, and I almost fainted that someone published the structure we just solved. That's how similar they were, right? Yes, Wally.
If you alter the dipole moment, do you alter like KCAP, AM?
The question is the question is if you alter the dipole moment, do you change the KCAT, KM? It's a really good question, right? These experiments were done by a group at the Israel Institute of Biological Sciences, Nest Siona. And in fact, they mutated 7 of the negatively charged groups on the northern hemisphere. And to our amazement, it had very little effect on the catalytic rate so far. And it's not clear yet how that can be reconciled with this argument that it's attracting it in. It's possible that under physiological conditions with different a number of differences may occur from what they're doing in vitro. But so far, it doesn't appear to be consistent, Wally. Bill?
Five similar structures have similar dipole arrangements?
Oh.
Have gorges or anything like that?
The question is, do these 5 similar structures have similar dipoles or similar gorges? No. This is what was interesting. Only the carboxypeptidase 2 has a similar gorge, but doesn't have a similar dipole at all. What I did do, and I can show you this later, we've made models of all of the other known sequences of acetylcholinesterase based on these coordinates, and those dipoles line up almost identically. Although there's only about 50% sequence identity for the different sequences, the distribution of charge groups is conserved. So it looks to me, despite these experiments that show the K-cat hasn't changed, something is preserved with these. Yes?
What's the influence of water on this effective dipoles?
Is there still dipole? Yeah, no, there is. I mean, it's the effective dipole is maintained, and it was measured by a Dieter Porsche in a number of different ways, and it's preserved. If you change the ionic strength, you'll affect it, of course. But in solution, the dipole is maintained. However, I should point out, the dipole is for the monomer. The way the 2 monomers form a dimer, the dipoles are exactly pointing in the opposite direction, and they virtually cancel out. So there's no effective total dipole for the dimer, okay? But locally, if you look at each individual one, you'll see a clear dipole as you get close, but from a distance, you'll lose it. Yes?
General question, you do the crystallographic structure on single crystals of a material, and you put them in the body or in situ, they're not crystalline in nature. I know it's probably a common question, but are there any changes in the function when you break up the periodicity of the crystal structure?
The question's common, but it's really an important question, okay, anyway. The question is there a difference really in the crystal structure and in solution, I think is what you're asking. And the answer is that, in general, it appears there isn't. And this can be checked initially by means of comparing crystal structures to structures that have been determined by means of NMR, which are inherently solution structures. However, there are cases, especially for rather short protein molecules, 50 or 60 amino acids, where they aren't large enough in some sense to make their own local environment. And those seem to be much more affected by crystal packing forces than ones which are larger. So in those instances, one has to be somewhat more careful, at least to look at the structures under different crystal packing arrangements. For these and other proteins, one can see inhibitors that bind. In fact, many proteins are active in the solid state and do their catalytic reactions. So in general, if there are large enough proteins, it appears that the crystal structure is really close to the solution structure. Yes, Steve. Oh, I don't know about that, Steve. I'm not certain. Okay. No. Now it's important based on what we've seen to check this. Yes.
Is the dipole moment of the monomer large enough that if you work at an isoelectric pH, you might be able to use an electric field to orient the molecules in solution?
These are just the kind of experiments that a new graduate student is doing, things like that. In fact, he's looking at fluorescent dyes that are being attracted to this. And I think it might even be a way, just using what you're suggesting, to maybe grow better crystals. Okay, so I think, yes.
Even in solution, you could get some spectacular NMR splitting because the dipolar interaction wouldn't be averaged to 0.
No, I think it would be really worthwhile. The only problem is that this is a large molecule, 65 kilodalton.
With a large
Well,
one, splittings could be, kilohertz.
this would be really interesting because for us it might be a way in parallel to the Lowey experiments to start looking at some of the dynamics, because that you can do more easily with NMR. Maybe we could talk afterwards.
There's one question way from the back.
I'm sorry. Yes?
People use very widely DNA sequencing to look at species divergence in evolution. From what you're saying, is it possible, will it then be possible to use protein structures and proteins which have, as you've said, quite different
question is, can one think about using three-dimensional structures for, in some sense, studying evolutionary relationship of molecules? I think the answer is, in general, yes. The only caveat that one has to worry about is that sometimes two proteins may fortuitously have a similar three-dimensional structure. In this instance, we think it's not the case, primarily because the catalytic residues are all lined in a similar place in the topology. But very frequently, some rather complicated folds appear to be just examples of just converging evolution, where different molecules have just converged to that same structure with no apparent relationship by evolution. Because if one looks at the active site on these, the active sites are in very different places on those kind of three-dimensional folds. And it looks like almost these folds are just simple extensions of alpha helices or beta strands in a more complex way. So if one can distinguish convergent versus divergent evolution, I think the answer is yes. Is it clear?
I think if you have any more questions, you can probably talk with Joel out at refreshments out in the lobby afterwards.